Welcome to my YouTube channel. I am excited for you to be here today. I am Sharoon Daniel and we will continue our walk through our past papers, and today we will discuss further questions of Foundational Paper 2 GCSE Biology AQA exam. Let's begin, and if you like my content, please like, share and subscribe for more videos and resources. Now let's begin with first question, which is question 5, and it is all about decomposition and decay. Here examiner is interested in checking your knowledge rather than just the memory recall. So let's start with first part of question 5, which is 5.1. Let's read this question together, and it asks to name one other organism which decompose and decay, and the answer is fungi, along with bacteria. These are the main decomposers. And now let's take a look at next picture. Figure 8 apparatus is shown, and the question is 5.2. What is the dependent variable? And the answer to this question is the pH of the milk. I will give you an exam tip that the dependent variable is the one you data collect measure. The independent variable is the one you change. Temperature. Similarly, next part of the question 5.3 is about control the temperature to specific temperature. And answer to this scientific question is put the beaker in a water bath. Exam tip here is that the water baths are the standard way to keep a solution at a precise, constant temperature in biology. Next part of question 5.4 asks you what type of acid is produced by digestion of fats in milk. And the answer is fatty acid. Exam tip. Fats, lipids, are broken down into fatty acids and glycerol. The production of these acids is why the pH drops as milk spoils. Figure 9 shows a line graph, and the part of question 5.5 asking you scientific reason why at 20 degrees pH does not fell under 4.5. Answer. All the fat had been digested. Exam tip. When a graph levels off, goes flat, it usually means the reactant has been used up or the reaction has finished. Now moving on to next part 0.56, which asks you one reason why rate of digestion is faster at 30 degree rather than 5 degree, and the answer 5.6. Reason for faster digestion at 30 degrees Celsius. The primary reason is that enzymes and substrate molecules have more kinetic energy at higher temperatures, up to the optimum. The reason, as temperature increases, molecules move faster. This leads to more frequent successful collisions between the lipase enzymes and the fat molecules, resulting in more enzyme substrate complexes being formed per second. Hence at 30 degrees is the optimum temperature for enzymes to work at maximum level. Next question required you to read graph in figure 7 at 30 degrees where the pH of the milk changes from day 2 to 3 according to the question part 5.7 to find out rate calculation, and the answer is the pH at day 2 at 30 degree was 6.0. The pH at day 3 was 4.7. Now let's calculate, and the fall in pH 6.0 to 4.7 equals 1.3 pH units day, in the final answer. Final question, which is 5.8, is about comparing rates, as this question asks for a ratio. You are comparing your answer from 5.7 to the given rate of 0.1 pH units day. The formula for this calculation is rate at 30 degree divided by rate at 5 degree. Example, if your answer for part question 5.7 was 0.4, the calculation would be 0.4 divided by 0.1 equals 4. You would write 4 times faster. Let's begin our next slide showing figure 10. Here you could see the details of this figure showing filtration happening in kidneys. Now you could see the blood contains plasma where water, salts, glucose and protein molecules are found. Let's see the filtrate where some contents of blood are filtered out as filtrates through the renal filter. Now keeping this diagram in mind and its details, let's address the first question, which is 6.1, and it asks you to match the given molecules to draw line to match the option given in front of them. Let's use physiological reasoning using the figure 10 to match the filtration movement of first glucose line draw to the molecules are small enough to pass through the filter. Then move on to second option protein line draw to the molecules are too large to pass through the filter. Here let me give you a GCSE exam tip. Remember, big stays, small goes away. Actually, small things like glucose and urea get filtered out of the blood, while big things like proteins and red blood cells stay in the blood. Next question is about asthma regulation, water regulation. Function of kidneys as they control water volumes of a person daily basis. Hence figure 4 shows data of a person showing water filtered from blood and water lost in urine. Next question is for 1 mark and examiner requires you to show your mathematical skill to calculate water reabsorption from kidneys in question 6.2. Let's dive into the answer to this question by carrying out calculation, as data in figure 4 shows that water filtered from blood is 160.0 decimeter cube, and if you minus 1.9 decimeter cube water lost in urine, you will get the answer, which is 158.1 decimeter cube. Now the exam tip for this question is this fact that most of what the kidney filters is actually put back into the blood, reabsorbed. Only a tiny fraction becomes urine. To help you ace these GCSE biology questions, I've broken down the answers according to the typical marking criteria and added specific tips to help you secure every mark. Next slide shows figure 11 where a patient with end-stage kidneys disease is to get treatment, and doctors have 2 options in front of them, either dialysis 3 times a week or kidney transplant. Figure 11 tells details about both treatments there. cost and how they will affect the patient in short term and long term. Now, keeping all this information in mind, examiner don't just expect you to know physiology of kidneys and their diseases, but they expect you to analyze the data and evaluate the statement to give your opinion about if doctor's statement. Let's begin with the question 6.3, which asks you to evaluate the doctor's statement. Note this is a 6 marks question, so please read and explain the answer with detail. So here is the answer for an evaluate question. You must give advantages and disadvantages for both options and provide a conclusion. First, arguments for transplant. Advantages: lifestyle impact, no need for time-consuming hospital visits. 3 times a week for 8 hours. Diet changes, no need to restrict protein and salt levels in food. Cost of procedure much cheaper over the long term. Transplant costs £5,000 year after the first year, compared to £35,000 year for dialysis. Arguments against transplant. Disadvantages: surgical risk are present, such as use of general anesthetic can cause damage to other organs. Immune system compromise, as patient must take immunosuppressant drugs for the rest of their life. Availability and longevity: the kidney may only last about 10 years and there is a shortage of donors. Conclusion: a transplant is generally better as it provides a higher quality of life and is more cost effective for the healthcare system, though it involves higher initial surgical risks. Here the exam tip is to use comparative language. Don't just say dialysis is long. Say dialysis takes 24 hours a week, whereas a transplant allows a normal daily routine. Now moving on to next question, which requires your math skills as it's a calculation question. 6.4, as it asks you to calculate the total cost, and note it's 3-mark question, so be careful with your data entry and calculation steps. So let's begin. As you need to calculate the cost over 5 years. The calculation: year 1, £17,000. Years 2, 3, 4, and 5. 4 years, £5,000 per year equals £20,000. Total cost: £17,000 plus £20,000 equals £37,000. Total cost equals £37,000. Exam tip is always show you're working. Even if your final answer is wrong, you can often get 2 out of 3 marks for showing the correct method. For example, showing that you multiplied £5,000 by 4.